Hello, biology, and welcome to your last new content of the semester in which we are going to talk about homeostasis. Now, we learned in our first unit that a biological niche describes the specific abiotic, non-living, and biotic living conditions that are really necessary and often required for life to occur. The best way we illustrated this was looking at certain types of soils or certain types of temperatures that trees thrived in, or certain types of trees that woodpeckers needed, or more specifically, if we looked at the pikas needed 70 degrees or colder ambient temperatures for them to live. But a biological niche is also seen at both the cellular and the tissue levels when it comes to the parts of our body and what they need. This might be oxygen, it might be temperature. There's a lot of things that the cells in our body require for life to occur. So what is homeostasis? Homeostasis is just what those things are. It refers to the ideal conditions for a particular variable that life will thrive in. And it's very particular for each type of cell or each type of organism, but there's always a particular range with a min and a max value that life is going to either survive in or thrive in, much like the fundamental niche of an organism. When our bodies leave homeostasis, it often creates damage. It's often not good for cells, for tissues, for the organism. It can often be fatal. It can result in death. If you are deprived of oxygen for a variety of time, a couple minutes, you will almost certainly die or have permanent brain damage. We control these parameters, given that homeostasis is so key, with systems and feedback loops using proteins between our cells and within our cells, which is why we care about proteins so much. So let's just look at a variety of homeostatic variables that our body requires. Excuse me. For example, the oxygen saturation in your blood, meaning red blood cells carrying oxygen, has to be above 95%. So 95% of the red blood cells in your body at any given time is carrying oxygen. In terms of the opposite, CO2, which is the waste product of our cells, it can only be below 29 molecules per nanoliter, which is a millionth of a liter. So we have a very tight range of the waste products in our body. And that's because CO2 can be really damaging to our body in high amounts. In terms of energy, we have a really tight regulatory system that keeps our blood sugar levels high to make sure that our body is always getting energy throughout the organs and muscles. Our internal temperature needs to be between 97 and 99 degrees Fahrenheit to keep our body in a range that is conducive to life. And even more importantly, we can see homeostasis when it comes to certain hormones or proteins. For example, testosterone needs to be above 300. Can be as high as 1,000 in males and between 15 and 70 for females, but this is very dependent on age, time of life, time of year, time of month. This, though, is regulated. Below this value or above this value often has compromises. So, how do we maintain homeostasis? One way that we maintain homeostasis is using feedback loops. And feedback loops is where cell signaling occurs between different cells that communicate with each other about what the body needs. And a feedback loop often is broken down into 4 parts. There is a stimulus, a detection, a control, and a change. So negative feedback loops occur when a change from a signal feedback system directly causes the stimulus to diminish in a way opposite of what the change was. And another way of putting it, as a little bit wordy, the product or the outcome of our negative feedback system loop changes the stimulus or symptom in the opposite direction. So it's a course correction. You steer to the left, so then you steer to the right. You go up, so you go down. You spend too much money, so now you save a lot of money. You're doing something that reverses what the stimulus or symptom was. So let's look at an example of that. your body temperature drops below 37 degrees Celsius, which is about 97, 98 degrees Fahrenheit. You are becoming hypothermic. You are getting too cold. Your body is going to detect that, where muscles are going to be released from your muscles and nerves, and it's going to travel to your brain, which is where you have your thermostat for your internal body temperature. So your muscles are going to be sending stimuli to your brain, telling your brain that, hey, it's too cold in your bicep, it's too cold in your quadricep. Your brain is then going to have a organ that releases more hormones, more proteins that are going to go and target muscle cells in response. So we're going to send information up to the brain, and then the brain is going to send information back. And that change is going to be your body starts to shiver. Your blood starts to pump harder in more to the core. We are going to do things to raise your temperature. So negative feedback loops are used to maintain homeostasis or get back to that steady state, constant, equal environment, that not too hot, not too cold, just right condition. It maintains it because we can go above or below what we need for homeostasis, and either way we can do course correction. So let's take a look at this. If we're looking at equilibrium or homeostasis for our body, if our stimulus is going below what equilibrium is, we're going to be using an increasing amount of detection, which is why these stars get bigger, to tell our body, hey, we're getting really cold, or hey, we're getting really hungry, or hey, we're getting really tired because we have not slept enough. As the stimulus grows, so too does your detection. At a certain point, you're going to reach a threshold. You're out of breath, so you stop sprinting. You stop and eat food because you're so hungry. You drink water because you're so thirsty. The control is going to be the way in which you reverse the trend of the stimulus that's impacting your feedback loop. And then we have the change. Your body begins to shiver, you pump blood to the core, and you return back to equilibrium. And the same thing can happen if you go above your equilibrium. Note that the pitch or the angle of the line can be either shallow or severe depending on the intensity. I can do a really fast 30-second sprint and run out of air, so I stop, or I can do a 2-minute interval running and still have the same effect. I'm feeling out of breath. And we see negative feedback loops in a variety of ways. We see them with blood sugar and glycogen storage. Your blood sugar goes up, so you store more glycogen, or your blood sugar is too low and you release sugar from your muscles. Your body temperature, which we just talked about. Your blood oxygen and respiration rate, you breathe more when you need more blood in your body. Hunger and satiety, so you eat when you're hungry. When you're full, you are not hungry. And also prey and predator populations. When prey is high, predators will go up. When predators go up, prey go down, which then results in predators going down as well. Now, a positive feedback loop is just when the effector basically creates the stimulus to increase in an exponential direction. It's a reinforced feedback loop. So the change in the stimulus will result in more of the stimulus. So we can see this with the perfect example of the zombie apocalypse. A human becomes a zombie. It's infected. They are infected by being bit. The zombie then can detect humans when they move or make sound. A zombie can infect a human by biting them, which results in more humans being bitten, so we have more zombies. This is what a feedback loop looks like. Notice there's not blue and green because the stimulus is the stimulus, which is the stimulus, with an increasing amount of detection and then the control, the thing that is causing more of it to happen and more of it to happen. We see this with uterine contractions during birth. We use a hormone called oxytocin to make babies come out of the womb. And as oxytocin increases in female bodies, uterine contractions increase, and increased uterine contractions make more oxytocin be released. Invasive species, in terms of their introduction and die-offs, more invasive species will result in a larger and larger population. Infectious diseases, one person comes back from break sick, then 2, then 4, then 8. And again, every time the control mechanism is an increase in the stimulus. And these are what examples of positive feedback loops look like. Exponential growth within a population. So this is our selected type 3 populations that grow really fast. And then also the tumor cells, or the size of tumors in cancer, where over time tumors get much larger over time because cancer divides at an exponential rate. And then our last thing we have to talk about is cell signaling. And cell signaling is just 3 patterns in which cells communicate with each other using hormones or proteins. And there's many individual types of cell signaling for each cell in our body, but the general structure all looks the same. So the first is reception, and this is where a hormone protein binds to a specific signal or receptor protein in the cell membrane. So this is outside of the cell, and this is within the side of the cell, and here's our phospholipid bilayer. So reception is just when a protein binds to another protein, and it causes them to interact, engage, be activated. This is very much like you playing tag with someone and you get tagged. So the reception has now occurred, you're going to be tagged. Or more importantly, if you're thinking about another analogy, it's time to eat. So you are interacting with a cookbook to find a recipe to make. The second aspect of cell signaling is called transduction. And this is completed when the receptor and the signal protein activate other proteins within the cell. And those proteins inside of the cell are known as secondary messengers. And this is really where we play the game of tag. So one protein is going to then use energy to activate another protein, and then that protein will be turned off. And then this continues as a cycle, where each protein in the chain of events is activating one thing after another. First, if I'm thinking about my recipe, okay, I have found my recipe. Step 1, step 2, step 3, step 4. These are all the steps that I'm completing sequentially to be able to create my recipe. And each of these is a specific protein that's using energy to activate the next in the series of events. And these brown clouds represent, once number 3 activates number 4, it becomes deactivated. 1 activates 2, 2 activates 3, 3 activates 4. It's a sequential pattern of activation. And then the last aspect of cell signaling is the last secondary messenger enters the nucleus and tells the cell to read some DNA and make a protein. So it might be a protein that is going to be used to respond to a change in the stimulus. It might be a protein that makes a physical product. It's going to be a protein that does some sort of work. And that's going to be where we have these proteins bind to our DNA, read that messenger or read that information, and then make it. So this would be where we're putting all of our ingredients together with the final recipe and making our food product. So again, all of this is dependent upon, if we go back in time, different types of proteins. We have our receptor proteins and signal proteins that allow us to communicate with each other. We have transport proteins that allow things to come inside and outside of our cells. And we have enzymes that allow us to break things up or down. So we're going to be putting all of these pieces together this week and after the midterms as well, but that is where we're going with this information. I hope this has been a useful video. We'll talk more about this on Wednesday and Thursday. I will see you soon.